Right, topic B9, biological pigments. It's going to look at some biological molecules and maybe try and understand why they're coloured. Previously, the colours that we've met have been in transition metal complexes. You see here's my transition metal ion, my ligands, and we were talking about DD transitions. So we've got splitting of these d orbitals due to some of the ligands pointing directly at the orbitals and some pointing in between. And if my electron is able to be promoted up, then that will be absorbing light, and if that absorbance happens to be in the visible spectrum, then the light that's left over is what I see as a colour. So for example, here's my copper sulfate, some light's coming in, it's absorbing kind of in this area, which means we're seeing colours more in that area. And we used a colour wheel to explain this. As a general guide, if something's absorbing here, we're going to see that kind of colour. So the factors that affect the colour is essentially asking the same question as what affects the size of that gap between those d orbitals. That difference is important in determining what wavelength of light gets absorbed. And what can affect the gap? Well, it can be the identity of the metal ion, it can be the oxidation number of the metal ion, it can be what ligand we've got, and also the shape. So if we think of the ligand, you can check in your data booklet, some ligands are called stronger field ligands and others are weaker, and if you've got a stronger field ligand, that's going to give you a bigger splitting, and with that bigger splitting it's going to be a larger energy, which means it's going to absorb at a higher frequency or shorter wavelength, and as a result you can deduce the difference in colour that you're likely to see. A factor that's going to be important for us here in determining colour, however, is the degree of conjugation. And that conjugation is arising from alternating single and double bonds. And it gives us something called a chromophore. Right? And these chromophores are able to absorb UV or visible radiation. And as a result, we're likely, particularly if you're absorbing visible, we're going to see a colour. So here you can see some chromophores, and you've got a conjugated pi electron system. Right? These pi electrons, these overlapping p orbitals, make the electrons delocalized across them. So if you think of what our double bond looked like, remember you've got that sigma head on, but then you've also got our pp, and that gives us a pi bond like that. And what we're seeing is that they are delocalized. So there's also overlap there and there. So conjugation, more conjugation shifts the absorbance to longer wavelengths. You see some examples here. This one here is obviously more conjugated. The absorbance is at a longer wavelength, giving us a different colour. And this idea is also used in sun creams. So we've got some conjugation here. That's going to be absorbing in the ultraviolet region and protecting our skin. Similar idea for indicators, which you've used in acid-base titrations. If we take phenolphthalein, then over here there's not much conjugation. There's a little bit in the benzene ring, but very little, and so it's only absorbing in the UV and it's appearing colourless. However, if we add some alkali and we open that bond there, the structure changes, and now you can see that I've got this nice alternating system of single and double bonds running throughout the molecule, and I'm seeing much more conjugation. It's absorbing in the visible region, and as a result I'm getting a colour. So that's what chromophores are all about. So porphyrin rings, this is a general structure. It's a chelate, so it's able to form some dative covalent bonds to a central metal ion, for example. Could be in there. You've got these lone pairs on the nitrogen. And examples that involve these porphyrin rings, which we'll come on to look at: chlorophyll, hemoglobin, and cytochromes. So here's chlorophyll in plants. There's our porphyrin ring. So this is able to trap light, and that's very important for photosynthesis. And you can see that chlorophyll A is absorbing in this part of the spectrum, chlorophyll B absorbing light in this part of the spectrum, and this bit's not being used, and as a result leaves tend to be green. You can read a little bit there about how the trapping of light works. Oxygen carriers also have rings a little bit like this, and the oxygen carriers that we need to know about are hemoglobin and myoglobin. Hemoglobin most important for us as humans, and we've got iron in a +2 state. It's a quaternary structure. There's 4 subunits that interact, and it's worth noting that the iron doesn't change its oxidation state. So the oxygen binds and then can drop off again. It's a reversible process, and we describe it as being oxygenated, certainly not oxidized. Hemoglobin can carry 4 oxygen molecules, myoglobin can only carry 1, so each of these hemes in here, each porphyrin ring you can see like that, that's able to carry an oxygen. Hemoglobin's got these 4 subunits, but you can see here that myoglobin has a much greater affinity for oxygen, so it's actually able to steal it from the hemoglobin if both are present. Right, so at this partial pressure, I'd expect my oxygen to go over to the hemoglobin. So with these 4 subunits, hemoglobin is able to show cooperative binding, and that means that when subsequent oxygen molecules bind, they bind more readily. So as soon as one joins, the second, third, and fourth will quickly follow. And this binding is affected by temperature, pH, and carbon dioxide levels. So if we look at those, well, actually maybe let's start here. Let's look at this, the fetal hemoglobin. So we've got our baby inside its mum, and the fetal hemoglobin, as you can see here, has a higher affinity for oxygen than the mother's hemoglobin does. So as that blood comes into contact with the fetus, the hemoglobin in the fetus rips the oxygen off it and takes it. So that means that the fetus gets enough oxygen to grow. In terms of the effect of temperature, if we've got respiring muscles, they're going to be getting hot, right? So luckily, if we're at a higher temperature, the affinity for oxygen is less, which means that my hemoglobin will drop its oxygen. So when it reaches the hot muscles, the hemoglobin dumps its oxygen, and that oxygen can be used by the cells that need it. Similarly, if we've got those respiring muscles, they're going to be producing a lot of CO2, if you remember back to the equation for respiration. And that CO2, because it's going to dissolve up in the water, it's a non-metallic oxide, it's going to give us a weakly acidic solution. So it also lowers the pH, and again, it means that my hemoglobin is going to have a tendency to dump its oxygen. Cytochromes also contain these hemes, these porphyrin rings. They've got iron in the middle of them, makes it a heme. And they're very important for electron transport. So if you do biology and you've seen these pathways, then there's a lot of electrons need to be shuttled around, and cytochromes will be involved with that. These substances can be poisoned by cyanide, which is how cyanide acts to prevent your body respiring. And here we are seeing an oxidation state changing. I should have added, highlighted just back up here, that carbon monoxide is able to bind very strongly to hemoglobin, and that can obviously stop the oxygen being carried around and can lead to people dying of carbon monoxide poisoning. Carotenoids, you can see there a nice chromophore across here, and they're precursors to vitamin A, which is related to healthy vision, and they're also involved to a degree in photosynthesis. Lipid soluble because they've got a very long hydrocarbon chain. They are susceptible to oxidation, right? And that oxidation can be catalyzed by light, metals, hydroperoxides, and it's partly because of all these double bonds that it's able to undergo oxidation, and it can lead to bad smells. Final group, anthocyanins, cause plants to be pink, red or blue. Aromatic compounds, look at all these fused benzene rings, they absorb in the blue and green parts of the spectrum, so we start to see more reddish colours, and a good example of them is found in red cabbage. Soluble in water because of all these hydroxyl groups, lots of hydrogen bonding there, and their colour can be affected by pH and the presence of metal ions. So if you look at this, you've maybe used lower down in schools, red cabbage is a pH indicator, and you can see here changing the pH, changing the structure, we're changing the conjugation, and as a result seeing a different colour. So analysing these pigments, we can use the same technique that we use for analysing amino acids. The good thing here is that because they are already coloured, we don't need to use an additional dye. And so you can talk about starting off with your chromatogram, drawing a pencil line, putting a dot on it, and then letting the solvent run through that, and then by measuring the progress of the solvent front and the dot, you can work out the retention factor. Another possibility, same kind of principle almost, is using thin layer chromatography, and here my substance is adsorbing to the surface of the plate. Again, my solvent, the mobile phase, is moving up and it's separating out the components, and again I can work out RF values. Now a benefit of this one is that I can actually extract those dots, so it's a way of purifying what we've got. So this is adsorption because we're sticking, so thin layer chromatography, adsorption, sticking to that phase. This is more partition because the substance is actually kind of binding to the liquid that's trapped in the cellulose fibers of the paper. This slide summarizes again what we just talked about there with the chromatography.